Now that we've talked about how to create hypotheses, we have to talk about how to test those hypotheses. And to do that, we're going to talk about experimental design. And this is going to take us through a series of 4 or 5 lectures where we talk exclusively about the ins and outs of designing experiments. We're going to talk about a lot of different things, but before we get rolling, let's just do a quick recap. First, we talked about the null hypothesis, which is one where we hypothesize that there is no pattern, or that our proposed mechanism is not the pattern. The mission of science is to reject the concept of randomness, or essentially to reject the null. Failure to find evidence in support of your hypothesis does not require the acceptance of the null. We never accept randomness. As an order of principle, we never accept randomness, no matter what. Failure to find evidence in support of your hypothesis simply indicates that there is no evidence to support your hypothesis, and therefore we cannot, at this point in time, reject the possibility of randomness, but we do not accept randomness as the truth. Of course, we never accept anything as truth. Finding evidence in support of your hypothesis does not mean your hypothesis is correct. It only means that the data support your hypothesis. And we talked about the example where we had the loudspeaker and the alligators. And if we find that the loudspeaker sounds of the alligator cause alligator behavior, it doesn't mean that our hypothesis is correct, because we're not actually messing around with real alligators. We're just messing around with a speaker. Ultimately, we never find anything to be truth or fact or false or wrong. We only find that our evidence supports our hypothesis. That's it. We also talked about the p-value, which measures the probability that your observations would be found if the null were true, or in other words, if there is no pattern or if there is randomness. A p-value of less than 0.05 or 5% is very conservative and represents a balance between logistical feasibility and the desire for truth. And before we get going on the next lecture, I wanted to give you a demonstration of the p-value. To help explain the concept of the p-value just a little bit more, I want to run through a simulation with you. Suppose that we are interested in knowing whether or not deer, white-tailed deer, prefer to exist, I guess, in wetlands versus drylands. And so I have a forest. We go into a forest and we're going to measure the number of deer in wetlands versus drylands. And so this is my forest right here. You can see a green outline. And then I have my wetlands, which is your double-barred outlines here, your double-barred blue outlines here, and then you have your dry lands, which are your orange solid lines here. So we have 3 dry lands and 3 wetlands. We could, of course, have more, but I just didn't have room to draw all of them. And we go in and we measure the number of white-tailed deer that we find in all of these areas. And this is what the data looks like. This is where the deer are. Now, looking at this, you can see, hey, there's more deer in the wetlands, and there's fewer deer in the drylands. And so you would reasonably conclude, well, there's deer prefer wetlands. Except that's not statistics. Just eyeballing the data is not that. And maybe you just happen to walk into the forest on a day where they're clustered in wetlands. So let's be clear. This is what we actually find. We go into the forest and we actually find this. This is what we observe with our own eyes. But now we have to ask our question, what's the probability that we just found this by pure chance? When, let's say that the world is actually random and we just happen to find this by pure chance. Another way of saying this is, let's say that we collect all of that deer in the hand of God, and then we just roll it up, shake it up, and throw them back out there. And if things were random, this is what it would look like. And if we did that again and again and again, things would always look random. All right, so if the world were actually random, this is what we would be finding. There's an old saying that if you give a monkey a typewriter, eventually the monkey will produce Shakespeare. If you shake up the deer enough times, eventually you will find something like this, what we actually found. But the question is, how rare of an occurrence is it to have found this if we are just constantly throwing the deer out? How rare of an occurrence is it to find this pattern? In other words, what are the chances of finding this if things are random? If we took up the deer and we shuffled them around and we threw them out 100 times, all right? If we threw them out 100 times, if the chance of finding the pattern that you're seeing on your screen, what we found in the first place, if the chance of finding this pattern is less than 5 times out of 100 times of taking them all in and shaking them all out, then we would say the chance of you walking into the forest and just happening to find the pattern that you saw of more deer in wetlands is just too small. Nature must therefore not be random. Your data suggest that we can reject the null hypothesis of randomness. It's simply too small. Now, if you need to go back and go through this again, please do. I'm going to go through a quick example in Excel that will guide you through this, not necessarily mathematically, but at least with numbers. All right, so let's jump to Excel here. And what we're going to do is a bunch of trials. Now, here are my actual data. Now, I don't have 3 wetlands and I don't have 3 drylands. I have 10 dryland sites and 10 wetland sites. And I go in the forest and I measure the number of white-tailed deer in wetlands versus drylands. And I find 17 in one, 15 in another, 10 in another, and then I go to drylands and I find 9, 11, 14, 14, 11, 7, etc. Now, I collect all of these deer up and I shuffle them up and I throw them out. And I throw them out at random. Now, given enough times, we will find this pattern, right? Because like I said, if you give a monkey a typewriter, eventually it will produce Shakespeare. So if you shuffle these values up enough times, you will get this order to the data. Let's go ahead and shuffle things up 100 times and see if we can come up with this pattern at all, at least 1, maybe 2 times out of 100. All right, so let's do that. All right, I'm going to do a little bit of Excel magic here that you guys have not learned yet. I'm going to copy this over, and then I'm going to use a function called RAND, or random, to get me a random number. And I'm going to draw that random number down. Now, what I'm going to do is I'm going to randomly shuffle all of these values around. All right, so data, sort by, and I'm going to shuffle it by column E. All right, they're all random now. Now I'm going to go ahead and I'm going to do that 100 times. I'm going to copy these data over and I'm going to sort things by the random number here. I'm going to do that again. I'm going to sort it by the random number. And you notice that these random numbers are changing. That's actually because it's a volatile function, which means that it will change whenever we do anything in Excel. Those random numbers change. I'm not trying to pull one over on you. They just do that. And it doesn't matter because it's always random. So I'm going to keep on doing this for 100 times. And you're going to bear with me while I do it.
Look into my eyes, you will see what you mean to me. Such a heart, such a soul. And when you find me there, you'll search no more. Don't tell me it's not worth trying for. You can't tell me it's not worth dying for. You know it's true. Everything I do it for you. into your
Okay, now what I have done, just to be clear, is I have randomly shuffled the values all over the place, 100 different times. Remember, this is our collecting the deer up, shaking them up like a Yahtzee, and throwing them out. And we're going to see now, I know I said earlier that we're going to see if we can find this exact pattern once in all of these, but of course we're not going to find this exact pattern in any of these. What we're more interested in is whether or not we can see the average. All right, is the average here is 14.5 for wetlands. And now what we want to know, after we've shuffled all of these things in, what are the chances, let me write this out, what are the chances of finding the same average, 14.5 deer per wetland, all right, what are the chances of finding the same average number in all of those random trials? All right, and what are the chances of finding the same average number of deer for our drylands, which was, if we take the average, you guys learned how to do the average already, so average was 11. What are the chances of finding the same average number of deer, 11 deer per wetland, per dryland, in all of these random drylands? All right, well, let's do that and see what we can find. All right, so this, let's call this the wetland average. Let's expand this. All right, that's the wetland average, and now we're going to do the dryland average. These two data points are our, I can't say that, are our actual observed averages. Now, if we collected all the deer and we shuffled them up and we threw them back out at random, what is the average that we're going to find? Well, our first average for our first random shuffle is 13.2. Our average for the dryland is 12.3. We did not find 14.5 and we did not find 11. What are the chances of finding 14.5? What are the chances of finding 14.5 or greater than 14.5 in our shuffled wetlands? All right, let's go ahead and calculate the second trial average. Again, we're 11.3, and we are 14.2. We're not finding that average. Let's go ahead and do that for each one of these trials. Now, I don't have to start singing Bryan Adams again. What I can do is I can just highlight this and I can drag this out, because what's going to happen is it's going to change the reference cell. You can see it changes the reference cell, and I can just move this on over all the way here. All right, and right there I've collected all of the random shuffle averages, and I want to know, what is the chance that I found 14.5 deer or more in wetlands? All right, what are the chances that I found that? Okay, cool. Let's figure that probability out. All right, no math. We don't have to do any math. Let's figure out how to do this. All right, so what I'm going to do is I'm going to insert another row there, and I'm going to copy the headers here, and I'm going to move these down. Now, oops, it's not trial one. That should be like that. Now, what I have to do is I have to move this. I have to do what's called a transposition. All right, I want to take this from being laid out horizontally, and I want to move it to be vertical. All right, so what I'm going to do is I'm going to copy that, and then down here I'm going to right click, and instead of our paste as values, I'm going to use another paste as special function, which is called the transpose function. And I'm going to transpose it, and you can see it just took whatever was horizontal and made it vertical. I'm going to do the same thing for these, but first I have to change them to values instead of equations. Otherwise it'll try to paste the values. So I'm going to first paste it as special as values, and then I'm going to transpose each one of these lines next to the trials. Oops, that didn't work. Transpose, there we go. Oh, no, it still didn't work. There we go. All right. I'm going to do that for my drylands. All right, transpose the drylands. All right, let's go ahead and move this down one column here just so I can write in here trial number, and this is wetland and upland, and I don't know why that is white. There we go. All right, let's expand that. So trial number, wetland, upland. I don't need it to be that big. Now, to answer what is the probability of finding 14.5 deer or greater in the wetland, or 11 deer or less than 11 deer in the dryland, this should say dryland, my apologies. What we're going to do is we are going to convert, you know what, I'm just going to convert these to numbers here, and I'm going to drag these down. And what now we're going to do is we're going to sort this, data sort, continue sort. All right, and I'm going to sort this one too, in order from least to highest, from smallest to largest. Okay. Excellent. What are the chances of finding 14? How many times did we find 14.5 or greater in all of this? Well, there's 14.5. It looks as though we found 14.5 deer or greater, deer per wetland or greater, in 1, 2, 3, 4, 5, 6, 7, 8 of the trials. Okay? So in 8 of the trials, let's go back up here. In 8 of the random shuffles, we found 14.5 deer per wetland or greater. And then what about 11? All right, well, where do we find 11 or less? Look, we found 11 right here, 11, and then we found 11 or less deer per dryland, 1, 2, 3, 4, 5, 6, 7, 8. And again, so we say in 8 of the random shuffles, we found 11 deer per dryland or less. So we would say then, let's zoom in on this, we would say then that there is a 8% chance of finding 14.5 deer or more in wetlands, assuming randomness, assuming that the null hypothesis is correct. And then we would say there is a 8% chance of finding 11 deer or less in drylands, assuming randomness. Okay? Now, the p-value, we commonly accept a p-value of 5%, which means that in order for us to confidently say that nature is not random, we actually need a 5% chance or less, or a 5% chance or less of finding 14.5 deer. Now, if you didn't catch, well, how did I get 8%? Remember, I did 100 trials, and I did 100 trials for a reason, because it makes the math easy. If each one of these trials represents 1% of the total likelihood, how many times out of 100 did I find it? If I found it 8 times out of 100, that means that there's 8%, or 8% chance that I will find it. Right? It just makes the math work out easier. We didn't find 5% chance of finding 14.5 deer or more, and we didn't find a 5% chance of finding 11 deer or less, which means that we cannot reject confidently the null hypothesis of randomness. Well, if we go back to our statistics slides, to our slides here, all right, it sure does look, here's the original data right here. It sure does look as though the data is not random. However, remember that we have to safeguard ourselves from finding patterns where there really are no patterns. And so 5% chance or less of finding a pattern, if the null hypothesis of randomness is correct, tells us that we can confidently reject the null. We did not find that here. We found an 8% chance, which is near 5% chance. So in statistics, we might say there's a trend towards it, but we could not find a significant effect. I hope that this has helped you to understand a little bit about what the p-value is, but if you are still stuck, ask me during the next class, and we will try to go over it a little bit more. There are three types of statistical studies. All of them are considered to be experiments in the broadest sense. Manipulative statistical studies, observational or survey studies. The difference is one where we observe and or when we exclusively observe and don't interfere. Survey is usually when we ask something of somebody usually it's a human-based survey. And then theoretical modeling, or theoretical or modeling experiments where we create equations that predict how the world will work. Now, I'm not really going to talk about this last one, theoretical, because it's kind of beyond the scope of our course, but it's worth just noting that that is one type of statistical study, and I'll give you an example of it. Let's start with this one from the previous class. This seemingly silly one, but this was an actual experiment where researchers noted that cows all seem to face the same direction while grazing. Now, I hypothesize, this is just one of the many hypotheses, that cows face the same direction while grazing because they are aligning with magnetic north. Note my hypothesis has direction, right? They are aligning with magnetic north. If I manipulate magnetic north, will the cows rearrange themselves? This is a manipulative question. An observational question would be, are cows aligned with magnetic north in my observations? Now, you might have to observe many different farms in order for this to work, whereas in the previous question, you simply alter where magnetic north is on a couple of different farms, and you see if the cows change direction. And then a theoretical or modeling question would be, out of all possible variables, including the direction of the sun, including what types of plants are nearby, what types of insects, maybe what other types of animals are grazing, is the direction that the cow is facing best predicted by an equation that only incorporates magnetic north? So with that example, I'm going to leave the theoretical modeling out of this, and we're just going to talk about manipulative and observational studies. The vast majority of experimental studies that you come across are manipulative in nature. The reason why, or we'll get to the reason why in a second, manipulative experiments are those that alter levels of the predictor variable and measures the response of one or more variables. Now, in this case, when we say predictor variable, that's the direction of magnetic north. That's what we're focused on manipulating because that's our hypothesized mechanism. And when we say alter levels of it, we mean alters the strength or alters the different types of variation of where magnetic north is. Manipulative experiments are the closest types of experiments where we can get to cause and effect. We call the manipulation a treatment, and we call the variable a response. So my treatment here is this gigantic magnet and the direction of magnetic north. And the variable, the response that we're measuring, is which way the cows are facing. The limitations of manipulative experiments are many. They are powerful, and we'll talk more about why they're powerful in a second. But for one, logistics and cost. Gigantic magnets are kind of expensive. In other words, sometimes things are possible, but not feasible.
we would love to be able to manipulate where magnetic north is, and what you're staring at is a gigantic magnet. But getting these is kind of expensive, and most grants don't have the money to buy these gigantic magnets to simply observe which way cows are facing. Even if it were possible to manipulate magnetic north with this giant magnet, you would need to bring this magnet to multiple farms for replication. And we're going to talk all about replication in the next lecture. The second problem is that even though manipulative experiments do get us close to causation, it's important to understand that correlation still does not equal causation. Remember, we talked about how just because one variable is related to another one does not mean that that variable causes the other one. If we manipulate where magnetic north is on a farm and all of the cows align to that magnet, it does not mean that our hypothesized mechanism is correct. It merely means that we are closer to saying something about that magnet is causing them to do this. Suppose they did align with that giant magnet. How would you know that they are just not simply staring at the magnet because they're curious? Maybe cow curiosity is something that you should factor in. They're not aligning with magnetic north, they're just aligning with something that they're like, what the heck is that? Or maybe they're facing the magnet because there are humans around the magnet and they like facing humans. They're herd animals. This is a great hypothesis. Maybe they just like facing humans because they are herd animals. We say then that the presence of humans, or cow curiosity, would be quote unquote a confounding factor that you want to get rid of to better control your design. We're going to talk a lot about confounding factors in our experiments. But confounding factors are any factor that affect your response that you did not control for, that you either accidentally did not control for or you cannot control for. Sometimes you can eliminate confounding factors. For example, maybe we can figure out how to drop off that magnet without humans being around. Maybe we can disguise ourselves. I don't know how you would get rid of cow curiosity, but you can sometimes control for confounding factors. A third problem associated with manipulative experiments is due to size limitations and the demands of replication, which again, we'll talk about replication in the next lecture, we often manipulate things on a very small scale. We're not going to bring that giant magnet in. Instead, we're going to herd a bunch of cows into a small area and stick a medium-sized magnet somewhere. Okay? The small scale of most manipulative experiments does not really represent nature. What you're looking at on this slide is an experiment where we tried to manipulate grazing effects as well as plant diversity in a grassland. But we can't do this across an entire grassland. We have to create miniature grasslands. Well, this small scale is nice for experimentation, but it doesn't really represent nature. I do this, and I suffer from this critique a lot, because I create small lakes in order to understand how toxins like pesticides or microplastics affect communities in these small lakes. I can't go dropping pesticides into lakes. Instead, what I do is I create these small lakes and I manipulate these small lakes. However, reviewers often come at me and say, well, these are not actual lakes. And I say, of course, but you can't measure actual lakes. Yes, there are 1,000,000,001 factors that make a small little pool like this different from a large lake, but I can't just dump pesticides or road salt or whatever have you into large lakes to understand their effects. Well, it's a compromise, and we make those sacrifices. And then later on, maybe we go visit a lake and we say, hey, look, this lake has been contaminated with road salt. I'm seeing the same thing in this lake as I saw in my miniature little ecosystems. Maybe what I saw in my miniature systems is what's actually occurring in this lake. If we were able to manipulate actual ecosystems, we would want to dump pesticides into multiple lakes. This is a key point. You're not going to just dump pesticides into one lake. Similarly, you're not going to just dump pesticides into one of these artificial pools because, again, we're going to use replication, which we'll talk about. You want to be able to see the pattern occur in multiple lakes, and you can't just go dumping pesticides into multiple lakes. Replication is important. If you just dump pesticides into this one lake and see the effects, that's just one occurrence. You need to see something at least more than once, and seeing something twice is just intriguing, but seeing something 3 times starts to indicate a pattern. If I dump pesticides into these lakes and I see fish die in every single one of these lakes, I gain more confidence that pesticides are bad. But if I just dump it into one lake and I see all the fish die, I can't say anything else besides what's happening in this lake. It indicates that maybe something is going on with the pesticides, and it bears repeating to see if there's an effect, but I can't actually see if there is an effect until I replicate it. This is the same idea as rolling a 6 4 times in a row. If you roll a 6 once, it doesn't mean anything to you. If you roll a 6 twice, that's intriguing. If you roll a 6 3 times, maybe something's up, and then if you roll a 6 4 times, well, then something is going on. Just like the die is the same in all of the tosses, so too would we want our multiple lakes to be the same in all manipulations. Well, ideally, all of the lakes that we dump our pesticides into would be perfectly identical. However, nature doesn't work like that. You're not going to find perfectly identical lakes. The lakes that you visit and dump pesticides into are going to vary in altitude, in elevation, they're going to vary in depth, in shape, in climate, and this results in more quote-unquote confounding factors that we simply cannot control, or that we try to control through a lot of time and energy. So maybe you're asking, could we just manipulate this lake multiple times? In other words, every single year we dump pesticides into that lake and try to observe what happens to that lake. Well, if you're thinking that, great. Stay tuned for the answer to that question. It is possible, but it has other problems associated with it. The fourth problem of manipulative experiments is that manipulations often entail animals that are fairly easy to manipulate. I would love to be able to ask this question. I hypothesize that plastic pollution in the ocean reduces whale fertility. I would love to be able to answer that question because it would provide us with more incentive for not dumping plastic into the ocean. My manipulative experiment is that I will feed whales plastic and observe the number of offspring. Of course, this is both unethical and unfeasible. I can't feed whales plastic. This would be the most ideal experiment. Really, if you were to do any experiment, would be what you want to do, but you can't do it. So, alternatively, we might test our hypothesis on something that is smaller and breeds faster. For example, like this little tiny amphipod that's very commonly found in marine as well as freshwater systems. There are hundreds of different species of this. Anecdotally, my friend Ricky went into graduate school thinking that he wanted to study tiger behavior, and his advisor sat him down and said, "You're probably going to have a lot of difficulty measuring tiger behavior, and so why don't we start off measuring something smaller like a cat?" Or maybe we can do something like fish, but not tiger behavior, because tigers are difficult to study. And lo and behold, 10 years later, he's actually studying these little amphipods and their behavior. Not exactly his wish, but maybe he'll see a tiger one day. That's not to say that if you want to study tigers, you shouldn't do it. Absolutely. I'm just saying you can't really put a wild tiger into a cage. Now, we could opt to do an observational study instead. And this is where we switch gears from manipulative to observation. Maybe we measure whale fertility in areas where there is a lot of plastic pollution, and we measure whale fertility in areas where there is not a lot of plastic pollution. This is perfectly reasonable. However, I'm not really convinced that you are going to find a place on this planet without plastic pollution. So this is not really going to be possible without manipulation. So you might want to dump a whole bunch of plastic into one part of the ocean and measure whale fertility. Don't do that. That's not cool, bro. However, there is probably areas with, or are probably areas with more and less plastic pollution. So we can still ask the question. We're not going to find any areas with no plastic pollution, but at least we can ask, is there more whale fertility in areas with less and less whale fertility in areas with more? In observation experiments, we really don't know the direction of cause and effect. For example, in our plastic question, perhaps whales with lower fertility just like to breed near plastic. We don't know that the plastic is causing the whales to have lower fertility. We just think that maybe the whales with lower fertility like to breed near plastic. I don't know why this might be, but it's a possibility. Another example, maybe a little bit easier to grasp mentally. For example, I observe that deer are commonly found in forests with high densities of maple trees. I hypothesize, and this is actually more of a prediction, I apologize. Sometimes, just for the sake of education, I will make my hypotheses a bit more like predictions than what we discussed in class. I hypothesize that an increase in deer abundance leads to an increase in the number of maple seedlings because deer will eat everything else in the area. Deer will not eat, well, deer will eat maple seedlings, but it's not exactly their preferred diet, and so they'll eat a whole bunch of other things in the area. So to test this hypothesis, I measure forests with lots of deer and forests without lots of deer, and then I measure the number of maple seedlings in each forest. Now, given my hypothesis, I would predict that forests with lots of deer will have a lot of maple seedlings, whereas forests without deer will have very few maple seedlings. If I find evidence to support my hypothesis, there's no way of actually knowing whether my hypothesized mechanism was correct. How do I know if deer caused an increased amount of maple growth, or if there are simply more deer in forests with maples because they like hanging around maples? Maybe it's safer in forests with maples because maples provide more cover for them. We don't actually know if the maples and the deer, if the presence of deer cause maples. We just know that they're associated with each other. So in conclusion, manipulative experiments are better. They just aren't always possible. It is important, though, for me to take an aside here and say that manipulative experiments are becoming more and more possible in our wonderfully global world that we have today. Networking has brought people together to conduct parallel manipulations in ecosystems across the entire planet. For example, suppose that I am interested in knowing the effects of fertilizer in forests. I now have a network of, let's say, 50 different people, 50 different forest types all around the world that I can call upon. and ask, what is the effect of fertilizer in forest? Please, everyone go put fertilizer into a section of your forest, measure the exact same thing, and report back to me. I actually participated in a very similar study where I asked, how does different climates affect leaf decomposition along streams? And in the bottom right, you can see a map of every single site around the world that did the exact same study at the exact same time and took the exact same measurements all at once. We all communicated via email instantly. We all said, okay, ready, set, go, and we did the experiment. This is pretty cool. We weren't able to do this 50 years ago, but now we're able to answer these really impressively broad global questions. This allows us to manipulate in a global and meaningful way. Technological advances and creativity have also made previously unfeasible experiments quite possible. For example, there is this global warming mesocosm experiment in England, and I've actually tried to replicate this when I was studying in Michigan, where they put these kiddie pools, $10 kiddie pools from Walmart, into the ground, and then they ran power cables through them, to not exposed power cables, insulated, so that it would warm them up. And we can simulate a slight change in temperature through this technological study. Another example, if we would like to know the effects of climate change on forests, there is a forest in the Northwest called the B4Warmed study. B4Warmed, get it? Where they actually laid power cables underneath of the forest floor and artificially heated up sections of the forest to see what it looks like when the forest warms up prematurely each winter. And you can see, we see growth in this area. There's a lot of technological gizmos here, of course, because it's still difficult and we still need a lot of gadgets in order to make this work. But we're getting closer to being able to manipulate on a large scale. One great experiment is called the Free Air Carbon Dioxide Enrichment, or FACE. This experiment asked, how does pumping a bunch of CO2 into the atmosphere, like humans are doing, and affect tree growth? Each one of these plots of trees and those white rings have pipes in them that are pumping CO2 into that forest. They're essentially spraying CO2 gas into that forest and asking, how does the forest respond? And you can see that we've replicated it. That's really cool. We're able to ask these really important questions on a large scale through controlled manipulation. Manipulative and natural experiments can further be subdivided into something called snapshot and trajectory studies. These are also known, if you've ever heard, trading space for time or trading time for space. I'm going to leave those colloquial terms behind and just talk about snapshot versus trajectory. Suppose that you have multiple lakes and you're looking to see what the effects of pesticides are on this lake. Those lakes were either exposed to pesticides this year or not exposed to pesticides. So let's say that on this collection of 16 lakes, half of them were exposed to pesticides and half of them were not exposed to pesticides. In a snapshot experiment, you would measure all of those lakes at the same time. Ideally, as close together as possible. You would send a whole bunch of people out to measure these lakes at the exact same time. And you say, well, this lake was exposed to pesticides and we don't see any fish. This lake was not exposed to pesticides, we do see fish. You see this again and again in 8 of the lakes that were exposed to pesticides, no fish. 8 of the lakes that were not exposed to pesticides, fish. Okay? The advantages of a snapshot experiment is that it is a rapid experiment, fits within a grant timeline, and that's important because when you get money to do an experiment from the government or from some other organization, there's usually a timeline associated with it, that you have to do this work by a certain time. And you can make more generalized statements about patterns than you could if you were to just measure a single lake. Of course, the disadvantage with doing this is that all of the lakes are kind of different in shape, size, altitude, etc. Realistically, you're not just going to find that pesticides kill all fish. You're going to find that pesticides may kill some fish in some lakes and not that many fish in other lakes. That's really what you're going to find. No experiment is just going to kill everything out there. And if we do find that, we really need to report that to the politicians immediately. Well, that's not usually what we're going to find. We're going to find a lot of variation. And because all of these lakes are different, it's going to be difficult to say whether the decrease in fish was due to higher elevation or whether it was due to higher temperature. Oftentimes, pesticides are more lethal when it's hotter out. Sometimes higher elevation means that it's colder, and so the pesticides might have less of an effect. Maybe there's more plants in some lakes, and the plants absorb those pesticides. There's a lot of confounding factors in this kind of experiment. Also, snapshot experiments might require extensive traveling, which is costly. And if you can't get a team of people together to measure everything at once, environmental conditions might change between your visit to one lake and the next. Suppose that you didn't have access to 16 different lakes. Suppose that you really only had access to one lake to measure the effects of pesticides. You know that the lake was exposed to pesticides this year. A trajectory experiment would measure the lake over time, but only to understand the effects of pesticides in that lake. It is really important to emphasize that you will only be knowing the effects of pesticides in one lake. We hope that this lake is similar to all other lakes, but we don't actually know that. We only hope that we can extrapolate our understanding of that lake to other lakes around the world. But there is a problem with doing this kind of experiment. Samples that you take through time are not independent of each other. In other words, the next sample is going to be influenced by the previous sample. Let me describe to you what I mean here. Suppose that there was a pesticide application of a lake. Imagine whatever lake you want. There was a pesticide application in the year 2000 and 2001. In the years 2002 to 2005, there were no pesticide applications. You might be tempted to measure the abundance of fish in the years 2002 to 2005 and say, this is what a lake looks like when there's no pesticides. And you measure 2000 and 2001, this is what a lake looks like when there are pesticides. That should feel uncomfortable to you. And the reason is because the years without pesticide application are influenced by the fact that there was pesticide application in those lakes previously. You can still take these measurements, but you must recognize that it changes your question. The question changes from how are fish generally affected by pesticides to how do fish recover from pesticide application over time? You are not looking at a lake that never had pesticides. You are looking at a lake that once had pesticides and now does not. If you could measure a lake that never had pesticides, the graph might look like the one on the top. But without measuring lakes that never had pesticides, you could never know this. You would only know the data points that are on the bottom. So if you only measure a lake that had pesticides at one point and does not have pesticides now, you're not asking how do pesticides affect fish. You're asking how do lakes recover from pesticides. I just want to back up here and just stress something to you. When you are creating your experiments are never wrong. They're never done incorrectly. However, your experiments can only address a very specific question. And so the way that you do your experiment might not actually address the question that you intend to answer. And that's a really important point. A lot of students are tempted to say, I did my experiment incorrectly. You didn't do your experiment incorrectly. You did an experiment. However, your experiment just did not address the question that you wanted to ask. Trajectory experiments also suffer from something called temporal confoundment. Now, we talked about confounding variables as something where one variable that you didn't measure might influence the response that you're trying to measure. What if in 2002 there was a particularly warm year? Well, if that was the case, then these data points are now the result of pesticides as well as temperature. You are measuring things through time, and climate conditions change through time. As a result, your experimental measurements are confounded by something else that happened over time. Another example of temporal confoundment is something called a chronosequence, where plots are manipulated at different points in the past to measure how an ecosystem proceeds with colonization through time. So you take one plot, you fence it off, and you measure what's happening in that plot over time. This is the same thing as measuring one lake over time. If you only measure one forest over time, then all of your measurements in 1 year are influenced by whatever factors the forest experienced the previous year. If you had a forest fire in 1 year, all of your future measurements will be biased based on that forest fire. So let's go to burning forests. Suppose that you wanted to measure the effects of burning on a forest. You burn the forest, and you measure its recovery over multiple years. Well, you only burn the forest once during that first year. You wouldn't really use the 5th year of measurements as a control unburned forest. That 5th year is biased based on the reality that you burned the forest in the past history. It would be better if you subdivided the forest into plots that you burn and plots that you don't burn, and then you measure each plot each year thereafter, much like you have lakes that were never exposed to pesticides and lakes that were exposed to pesticides. And you might be saying, well, isn't one plot biased based on its proximity to another plot? Well, yeah, and we'll get to that in the next lecture. Don't worry about that just yet. So this is a compromise between a snapshot and a trajectory experiment where you subdivide your system into the plots that you want. So similarly, you could subdivide a lake into individual treatments and put pesticide in one of these yellow dots and not put pesticides into another yellow dot. However, as you might guess, these treatments are still not independent because they come from the same lake, and fish will swim between these yellow dots, and pesticides might drift. It's a little harder to think about fire drifting from one forest plot to another, especially if we control that, but it's a lot easier to think about pesticides drifting. So you can't really subdivide this lake and expect that those samples might be truly independent. Maybe if we figure out some ingenious mechanism of sectioning off a lake, which people have done, we could do that. A real-time example of this, Dr. Christine Thompson's lab is currently exploring how dumping old shells, I believe that she uses whelk shells and old oyster shells, that are seeded with living oysters. Small oysters will grow on those dead old shells. She's looking at how dumping old shells into the ocean can help to restart oyster reefs. Her lab examines water quality measures over the new oyster reefs, for example, water clarity, how much algae is there. And they compare it to sites that don't have any of those shells that they dumped in. However, she has multiple sites that have shells and multiple sites that don't have shells. However, it's all one ocean. And if the sites that they measure are too close to each other, they might influence each other. Algae from one site might drift into another site. And so the measurements that she takes, that her lab takes, might influence each other. Ultimately, all plots in whatever experiment you're doing are confounded with each other because they are all part of planet Earth. In an extreme example, suppose that you're measuring pollution over one country and you're trying to look at how pollution in China affects China and how pollution in the United States affects the United States. Well, pollution from China will drift over to the United States or vice versa, depending upon which way the winds are going. And so that's a problem. It's confounded, spatially as well as temporally confounded. So we compromise. No experiment is perfect, right? There's always confounding variables. And this is one of the reasons why experiments can never demonstrate anything beyond any doubt whatsoever, because there is no experiment in the history of the world that is ever perfect. You might think it is at the time, but time has always told us no experiment is perfect. So the key is that you have to have careful interpretation of your experiment to prevent overstepping what your experiment suggests. And this is where a lot of scientists mess up. They try to conclude something from their study that their study has no business actually saying. All right? So when you're thinking about your hypotheses and your predictions, please take into account, can you actually test these? And if you do test these, will it actually answer the question that you intend to answer?